Hello again, and welcome to our third lecture in the cardiovascular system, where we're going to be talking about the conduction system of the heart. So I want you to think about, what does conduction mean? If you recall insulators versus conductors, what can a conductor do that an insulator cannot do? And that's transmit electricity. So typically conductors include metals and water. Insulators could include rubber and glass. Conduction is the process by which electricity is passed through a substance, and electricity passes through our heart. But more than that, electricity is generated in our heart. It's a really cool tissue. And so you might have seen a movie or a show where somebody's been diagnosed with brain death and their body can be kept alive by ventilators. Or maybe you've just seen Indiana Jones and the Temple of Doom, and somebody's heart is ripped out of their chest and it continues to beat on their own. Well, your heart actually generates its own electricity that's separate from the brain in a series of nodes and conducting cells called the internal conduction system of the heart. As long as the heart has oxygen, it can continue to work. So you should have studied the nervous system. That was in 201. And so you might recall that the medulla oblongata of the brainstem regulates heart rate. So if the brainstem is damaged, the heart will eventually stop beating. Heart in a box is an interesting extension on the intrinsic abilities of our heart. Through this technology, we are finding ways to let a donor heart continue beating outside of the body until it is placed into a recipient body. That is crazy. So intrinsic means from within. So for example, you might be an intrinsically motivated student, where you're motivated from within just for the sheer sake of doing well, and you aren't necessarily pushed by external factors. Or maybe you are extrinsically motivated. Either way, you're in this class, so I think there's a little bit of intrinsic motivation going on. When it comes to the heart conduction system consists of nodes and specialized cells that work together to cause the atria and the ventricles to contract at precisely the right time. It's best thought of electrical wires that run through the heart. And interestingly, these nodes and cells are made up of specialized cardiac muscle tissue. It's not nerve tissue. So the heart does something pretty special. When you are learning about the nervous system and muscles in general, that in order to contract, the muscle has to have a nerve supply. And as it turns out, the heart doesn't need that. The heart muscles, they can contract on their own. And one heart cell is able to generate its own action potential without any stimulus. And we call this autorhythmic. In other words, they can contract on their own in a regular and continuous way. So once that action potential has begun and been generated, it's going to conduct itself down a series of what is best thought of as wires. But it's basically this conduction system. And each area is going to signal to the next area to fire off its action potential. So the signal will be conducted down in a rhythmic order. And the signal is going to start at the SA node. The SA node is located in the right atrium at the base of the superior vena cava. And this SA node has specialized cardiocytes that can generate their own action potential. And those cells are called pacemaker cells. So these cells will generate an action potential, and that action potential will begin to spread through the heart. So this diagram showing those yellow lines, that is showing the action potential spreading through the right atria. And then it will eventually cross over into the left atria. And this is what we're looking at when a doctor or a nurse puts leads on a patient, and then they are reading an ECG. Sorry, they used to be called EKGs. They're more commonly referred to as ECGs now for echocardiogram. And in this diagram, you're going to see these waves. So the SA node firing is at the beginning of this very first wave, which is called the P wave. So as the signal is spreading through the right atrium, it will eventually get to the base here, right above the atrioventricular valve, the tricuspid, and it will get to what's called the AV node, which stands for atrioventricular node. So located where the atria meets the ventricle, but it's going to be located a little bit more on the right side. And this is the electrical gateway to get to the ventricles. Remember that there is a fibrous skeleton that we talked about before that is preventing the electricity from traveling anywhere except for this conduction system. It acts as insulation, so the only way electrical signals can get there to get to the ventricles is through this electrical conduction system. So at the top is the AV node, and then there is a short branch here, and that is called the AV bundle. And the AV bundle used to be called the bundle of His, but it's more referred to as the AV bundle now. That used to be a person's name. And that AV bundle will split. So you can see a splitting of the bundle, and it will split into the left and the right bundle branches, which will travel down the interventricular septum. And then once it gets down to the apex of the heart, it's now going to do a U-turn, and it's going to go up the sides of the ventricles towards the top of the heart through what's called Purkinje fibers. And as a reminder, we talked about these gap junctions or electrical junctions that are found in heart muscle, and this will allow the signal to travel from cell to cell. So take a second to orientate yourself on this image. So we're going to locate the SA node right here at the top of the right atrium. And it's at the base of the superior vena cava, where those pacemaker cells are going to fire off an action potential. And it will travel down the right atria as well as across to the left atria. And when it travels down the right atria, you can see that the AV node, which is labeled number 3, is located at the bottom of the right atria where it meets the right ventricle. And then we have that small little branch, which is going to be the atrioventricular bundle. And that atrioventricular bundle is going to split into the left and right bundle branches, which go down to the interventricular septum. And on the left and right side, that's number 4, And then number 5, it goes down and makes a U-turn to go back up the sides of the heart. This is also the way that the electricity spreads. Electricity starts in the atria and it goes down the middle and then it goes up the sides. Remember, electrical contraction will follow electrical stimulation. Muscle contraction follows action potential. Since the electrical conduction system starts in the atria will contract first, and then the ventricle contracts. So it's a sequential contraction. So regarding nerve supply to the heart, while these nerves do not make the heart contract, they can make it contract faster or slower, or they can make it have a stronger contraction. In general, sympathetic nerves will dominate the control of strength of contraction. These nerves will originate in the lower cervical or the upper thoracic segments of the spinal cord, basically the part of the spinal cord that's near the heart. And they'll start in the lower neck or upper chest. And if you go way back in your learning to the nervous system, I hope that you maybe remember about learning about the sympathetic chain ganglia, which are right next to the spinal cord. One of those is a cervical ganglia. That is where the ganglion is. The postganglionic fibers will travel through the cardiac plexus, then continue as cardiac nerves to the heart. Your heart rate is strongly influenced by both divisions, but it's mainly dominated by the vagus nerve. The cardiac nerves will carry not only sympathetic efferent fibers, those are the ones that have an effect, but they will also carry sensory or afferent fibers from the heart to the central nervous system for interpretation. And these fibers are important in the cardiovascular reflexes and in the transmission of pain signals from the heart. So you have to remember that sympathetic nerves had 2 neurons in their chain. The first neuron was short. It had a synapse or synapses at the ganglion right next to the spinal cord. And then the postganglionic neuron would be the effector. So that's where we have the fibers of these cardiac nerves that will terminate at the SA and the AV node. And they also go into the muscle itself as well as other areas of that system. Let's remind ourselves of the nervous system. So find the picture of the heart, and then you can see that our preganglionic fibers come off of the spinal cord. And then they synapse right there in the sympathetic chain ganglion. And then the postganglionic fibers go through the cardiac plexus, and from there, the cardiac plexus, they will go to the heart. Okay, so we can start with the heart here. You can follow that. So the parasympathetic nerves, if we remember, parasympathetic is rest and digest. So these nerves are going to slow down the heart. And this is mostly going to be done by the vagus nerve. The pathway will begin in the medulla oblongata with the vagus nerve, since that's where the nerve is going to come off. Can you remember which number the vagus nerve is? Of course I'm going to tell you, but I want you to think about it. It's number 10. But cranial nerve 10, or the vagus nerve, will also go to the cardiac plexus. And it's also going to be part of those cardiac nerves. But unlike the sympathetic nerves, which will also go to the muscle cells, the vagus nerve does not. It doesn't go to the muscle cells. It simply affects heart rate, and it will not affect the strength of contractions. So if you look at the very top, you will find the medulla. And you can see that the vagus nerve is coming off. And it's going to go to the cardiac plexus, which will then end at the heart as one of those cardiac nerves. So let's put some of these events together, and when the heart contracts, we call that systole. And when the heart relaxes, we call that diastole. So even though systole and diastole can refer to any part of the heart, usually we're referring to the ventricles. So ventricles contracting would be ventricular systole, and ventricular relaxation would be ventricular diastole. So let's use terms and everything that we have learned so far. So ventricular filling is mid to late diastole. The ventricles are not contracting. The atrium is in diastole and the ventricle is in diastole. No one is contracting. It is at the end of diastole when the atria will contract, as we mentioned before. So this ensures that the atria are empty and that the ventricles are full. Now the atrium is in systole, but the ventricle is in still diastole. So now we are in the second phase where the ventricle begins to contract. Now the ventricle is in systole and the atria is relaxing and in diastole. At this point, all the valves are closed, and the chordae tendineae prevent the AV valves from popping open into the atrium. So, if you think, why are the pulmonary and the aortic valves closed? Well, think about what has to happen to open valves. They have to be pushed open by pressure in the ventricles and that blood pushing against them. There will be a phase where that pressure is building in the ventricles, but the pressure is not high enough yet to open those valves. There is also blood pressure on the other sides of those valves as well. There is blood pressure in the actual aorta, and there is actual blood pressure in the pulmonary trunk. So when blood pressure in the ventricles is greater than blood pressure in those vessels, that's when the valves will open. So if we think about isovolumetric, iso means the same, volume means volume. So what that is saying is that the ventricle is contracting, but the volume of the ventricle has not changed. And it won't change until the pressure is high enough. So when the pressure is finally high enough, and it's higher than the blood pressure in the vessels, then those valves will open and the ventricles will begin to eject the blood. During this time, the atria are also filling, and the semilunar valves will close, and the ventricles will relax, and all valves will close and everything will be back in diastole and we'll start all over from the beginning. So all of that was all for one heartbeat. The normal heartbeat was triggered by the SA node, and we call that the sinus rhythm. Well, every now and then you can get what's called an ectopic focus. So remember that the term ectopic means something is happening at a different location than it's supposed to. So if the SA node is damaged and it's not working, you actually have a backup pacemaker. That's called the AV node. And the AV node is capable of generating action potentials by itself. And the AV node can take over as a pacemaker. But instead of being at 70 to 80 beats per minute, it's only about 40 to 50, which is enough to keep you alive. If you lose the AV node, well, the Purkinje fibers will be able to generate their own action potentials, but they can only do it at about 20 to 40 beats per minute. And that's not enough to sustain life. Might be enough to, if you're with somebody, to call emergency and get emergency professionals over to you, but it won't be enough to sustain you. So we're going to do a deep dive here. And we have to dig our minds around normal physiology of muscle contraction. So muscle unit of 201. And also action potentials. So with action potentials, there was a depolarization of the membrane caused by sodium entering into the cell. And then there was repolarization when potassium left. And with muscles, we have a resting potential of about negative 70 millivolts. And their threshold for muscle contraction was negative 55 millivolts. Well, the first thing this SA node does not have a stable resting potential. It doesn't hang out at negative 70. It has a slow and continuous drift. And it's going to start at about negative 60. And it will have the slow inflow of sodium. And because we're slowly adding this positive sodium to the inside of the cell, we're going to have a slow upward shift. The negative 60 will get more positive very gradually, not sharp like what we're used to seeing. And this is called pacemaker potential. And here in hearts, we have the threshold that's negative 40, not negative 55. So as we have this slow drift, eventually it will reach that negative 40. And when that happens, we have a voltage gated and very fast calcium channel. And voltage gated means fast sodium channels open. In muscles, we only talked about the sodium and potassium. And now we're going to have to throw in calcium. So there is a third ion channel we will see in these pacemaker cells. Once the membrane threshold of negative 40 is met, sodium and calcium channels will open and we'll have a really sharp depolarization that's going to keep going until it reaches 0. Now we'll have regular repolarization. And calcium channels are going to open, so calcium will leave the cell. And the cell membrane will start to head back down and get more negative again, which is called repolarization. Then we will start all over again. And this pattern will occur about every 0.8 seconds. And that gives an average resting heart rate of about 75. Looking at this chart, there are 2 beats on this chart. And notice, starting at negative 60 millivolts, we have that pacemaker potential and that slow inflow of sodium. Slowly, it's going to get up to a threshold of negative 40, and then you have sodium and calcium channels both open for a very fast depolarization. Then we'll have potassium start to flow out, and that is the repolarization. So the signal from the SA node will stimulate the 2 atria to contract almost simultaneously. It's going to reach the AV node in about 50 milliseconds, and the AV node is going to slow that signal down. And it's going to make it take a little longer to get from cell to cell. So in the AV node, the cardiocytes will have fewer gap junctions in between them. So that way that signal will be delayed and it'll take longer to get to the next cell. And it delays the signal by about 100 milliseconds, which is just enough time to allow those ventricles to fill. So I just realized I have a typo on here. This is supposed to be spiral, S-P-I-R-A-L. It's okay. So the signal, once it gets through the AV node, it's going to travel very quickly through the AV bundle and then through those Purkinje fibers. And essentially, it's going to reach all of the myocardium almost at the same time. So the myocardium depolarizes and contracts almost in unison. Those papillary muscles will get hit slightly later. I mean, very, very slightly later, just because it is a further distance. Ventricular systole will progress up from the apex of the heart. And that spiral arrangement of the myocardium twists those ventricles slightly, like bringing out a towel. So you can pause this here and try to identify these structures and test yourself. And I'm going to keep moving on. So here are the answers. We want to make sure we can identify all of these structures and their functions. So remember that the SA node is the pacemaker of the heart. It creates that initial impulse to start contraction of the heart, firing across the atria and causing them to contract. The AV node will slow down that signal, delaying the impulse so the atria fully have time to empty the blood, and the bicuspid and tricuspid valves will close. The AV bundle will carry the impulse towards the ventricles, and the bundle branches will carry the impulse to each separate ventricle. The Purkinje fibers will spread through the ventricles, causing the ventricles to contract. So for the next few slides, I'm going to try to use color coding to keep things organized. And I'm going to use the color purple to show when the atria and the ventricles are contracting, and to denote when the atria and the ventricles are relaxing, I'm going to use the color yellow. Another color coding thing that I'm going to be doing is I will have the title of the segment or the wave in a color, and then I will have that segment of the ECG matching. So that way you can see which segment matches the ECG. Okay, so the first segment we're going to talk about is the P wave. And in this wave, we have the SA node firing. It is the first wave you see on that ECG. And that SA node is a special group of cells known as the pacemaker of the heart. These cells will set your heart rate. The SA node generates an electrical impulse which spreads away from the SA node through the right and left atria. This is why we see a blip on the ECG. The bump up is the electrical activity of the SA node spreading through the atria. Electrical impulses cause tissue to contract, and the atria contract, forcing their blood into the ventricles. And a little fun fact, the impulse stays only in the atria due to that band of tissue in the heart. That's referred to as a non-conducting band that surrounds those valves and keeps them from contracting. So during the PQ interval or the PR interval, this is when the atria have finished contracting, but blood is still dumping into the ventricles. And the bicuspid and tricuspid valves are currently open. So note on the diagrams in this slide, it does not show the actual activity of the valves, but it's focusing on the depolarization and repolarization of the chambers. Also recall that the AV node will delay the impulse each time your heart beats. This is why the graph shows a flat line for a moment, no electrical activity. Well, this is important because this delay allows the atria to fully empty of blood and the bicuspid and tricuspid valves to close, preventing the blood from flowing backwards to the heart. So now we'll look at this second complex, which is the QRX complex here. And this is ventricular depolarization. And so I want you to note the apex of the heart. So during this time, the impulse will travel through the AV bundle into the AV bundle branches, which carry the impulse to the left and right branches to those ventricles. The large peak on the EKG or ECG is the moment the electrical impulse spreads through each ventricle. It takes a large impulse to contract these massive muscular chambers. The ventricles contract in from the bottom to the top motion. This forces blood out of the ventricles through the pulmonary and aortic valves. At the same time, the atria are going to begin to relax. And atrial repolarization is hidden on the ECG because it occurs roughly at the same time as the electrical signals generated by the ventricles. But those electrical signals from the ventricles is significantly greater in amplitude, basically masking the smaller atrial repolarization that's actually referred to as the TA wave or the TA wave. Once the ventricles have fully contracted and the electrical activity has paused momentarily, we are at the ST segment, which is here. And so we have ventricular depolarization is complete. All of the blood from the right ventricle is passing to the pulmonary arteries, to the lungs, and all the blood from the left ventricle is passing to the aorta, to the body. And then we have the T wave, which is this final wave here, and that is ventricular repolarization. which is also going to begin at the apex or the tip of the heart. And so during this time, the heart is resetting from the inside to the outside, which causes that bump on the ECG wave. And the ECG as a T wave. So now we have the heart that's been officially reset and ready to start again. The electrical activity is paused, but it is about to start again when the SA node generates an impulse and when the P wave occurs. The full cycle of the electrical activity of the heart lasts about 0.8 seconds. And this is a great diagram that is available in your book. I highly recommend to take a look at it or screenshot this. So when we have deviations from ECGs, they can detect disease of the SA or the AV node or the wires that make up this conduction system. Heart enlargement can be detected if the signals have further to travel than normal, so it'll take longer. And if you think about it, everything requires sodium, potassium, and calcium. So if your sodium and calcium are screwed up, then these electrical signals will be screwed up too. But what you can see here, and this is another image from your book, you have a normal sinus rhythm, and it's a steady beat. You can see all the waves, and there is a consistent time where we see each of these peaks. And so B is showing you ventricular fibrillation compared to the normal sinus rhythm. And so ventricular fibrillation, this is when there is chaotic electrical activity. The SA node and the AV node, the bundle branches, Purkinje fibers, all of those electrical signals are just going all over the place. And because electrical signals are traveling randomly, we don't have a contraction in such a way that the heart can expel blood. So the heart is not pumping blood to the body. And the coronary arteries are not getting their perfusion. So this will lead to a myocardial infarction because we are not getting any coronary perfusion or blood supply, and it kills very quickly if it's not stopped. The name of this fibrillation machine we use is called a defibrillator. And so a defibrillator is those things, you might have seen it on a movie where they're in an ER and somebody flatlines and they rub those paddles together, put it on the patient's chest and go clear, and then shock them, and the patient comes back to life, and you see the bumps go back on the ECG. So the goal of defibrillation, when you shock somebody with those paddles, is to stop the heart. You're trying to depolarize and reset the entire myocardium. So all the electrical activity resets and you can start over. What you are hoping for and counting on is that the AV node will start up and its normal sinus rhythm again. This will not cure muscle disease if you have it, but it can at least restore the electrical rhythm and give you time to get corrective actions and get to an emergency room. And I want you to note that on this ventricular fibrillation, there are no P waves and no QRS waves. And so here I have the normal sinus rhythm up top, and then I have an ECG of heart block and an ECG of premature ventricular contraction. So if we look at image D, heart block, notice that we have a P wave and a QRS wave, and then a T wave, and then a P wave. But then no QRS, no T, just another P wave, and then another P wave. And then finally, we have a QRS. The P wave indicates that the atrium is doing fine. It's contracting, but something's happening after atrial contraction. The ventricle's not getting the signal. So this is called heart block because something's blocking the signal from reaching the ventricle. Heart block is a failure in the conduction system to go all the way to the bottom or the apex. So that way we have normal atrial and ventricular contraction. So then we'll have atrial, then atrial, then atrial, and then finally we'll have a ventricular. The bottom of the page where I have image E, if you look at the arrow, it looks kind of weird. This is called premature ventricular contraction, or PVC. This set of waves did not conduct in the normal place, so it's a weird electrical signal that started on its own somewhere. It's an ectopic focus. So this could be some sort of irritation disease like inflammation or maybe a damaged ventricle. It could be, so this disease portion is setting off its own electrical signal, and that's why it's called premature ventricular contraction. If I didn't mention it, it's called sinus rhythm because it refers to the normal rhythm that originates from the sinoatrial node. So that's why it's called sinus rhythm. And in your virtual lab, we're going to learn about this. So notice that one of the heartbeats is coming much faster. If that heartbeat is coming much faster, then we use the term tachycardia. Tachy means too much, and cardio means the heart. So this is when the heart rate is too fast. And the opposite is bradycardia. Brady means slow, so this is when the heart beats too slow. And notice that those waves are going to come in a lot slower. So we have sinus arrest, which is when the SA node fails. And then the AV node also fails. No systole means no contraction. And when you see no waves, that means there's no electrical activity at all running through the heart. So sinus arrest will be detected on one of these ECGs by about 2 box lengths, and that is considered to be sinus arrest. And we have what's called a pacemaker, which can be inserted into the body, and there will be a lead that takes place of the SA node. So we made it. And next up, we'll talk about principles of pressure and flow.